we can get started at least with the announcements. So I have a couple of quick announcements before we get into the agenda for today on behalf of Marina and myself as chairs of this working group. But are there any other sort of general updates or announcements from anyone on the call that would be relevant to this forum? If not, so I just want to quickly mention that our next call, even though this is our first call for 2024, so welcome everyone to 2024, but our next call, which is currently scheduled for the 25th of January, will actually be cancelled because it's meeting free week, which is a quarterly event that we do try to observe within ClinGen. So that means our next meeting after this one won't be until the 14th of February. And the other thing I wanted to quickly mention is that the Biocurator working group within ClinGen is a forum for all of the biocurators within the ClinGen ecosystem to sort of connect. It's a training opportunity, education, a place for people to sort of connect with other biocurators. And the way that people become members of this group is that when you're a volunteer as a biocurator through any of the various expert panels, that the coordinator of that expert panel links you into the mailing list and to this group. We do know that unfortunately some people slip through the cracks, and so we are trying to make more of a conscious effort to make sure that people aren't falling through. So if you're a biocurator on this call and you know of someone else, either within your lab or somewhere that isn't part of this listserv, if you could ask them to reach out to the coordinator of whatever group they're involved in, or even reach out to us directly. So from the Biocurator working group page on the ClinGen website, just check in with Kazan, who will be able to get you linked in. But we will also be putting a reminder for this through like the ECT newsletter and various other forums as well. But just to make sure that we're not missing out on having anyone as part of this working group meeting and the education opportunities that are here. But with that, I will introduce Bill, who is one of the staff scientists at the UNC Biocuration Core, and is giving an update today on the Retina GCEP. So I will hand over to Bill.
Cool. Thank you, Alicia. Thanks for introducing, and thanks a lot for the opportunity to kind of share some of the things that we found with this audience. Awesome. Screen sharing is working. Cool. And hopefully everything is in view. I don't know. Right. So like you were saying, here just as a representative of the Retina GCEP, this group has a lot of really interesting pre-curations and gene curation findings. And so we just wanted to kind of share with this group, what we found so far. And please note that you can unmute anytime any of the participants and either ask a question or make a comment, or if you've, come across some interesting pre-curation issues, please feel free to put in the chat and we can maybe discuss together. So I just want to make a quick caveat that all the presentation findings and interesting things that I'm going to show, it's the work of a whole lot of different people. So this is a pretty big GCEP. Our chairs are Rob Huffnagel and Gavin Arno. They've been helping me to plan the presentation. I've been totally pirating their slides. And our group is coordinated by Christy Lee, and we have a co-coordinator, Blair Compton. We have an awesome group of experts that helps us every step of the way navigating a lot of really weird lumping and splitting issues. So if you're kind of a curator who's interested in lumping and splitting, this is just an awesome group to work with. And you can see we have a pretty good curator group as well. We have about 15 curators and these past members. So I'm showing the work of a whole lot of different people. So just like a quick thank you to them, and especially to Jenny, who has been helping me to kind of plan the presentation. Right. So the topics for discussion today, we were thinking maybe we could talk a little bit about the scope of the GCEP, the different number of genes that we're working on, and sort of like how we tie into other ClinGen groups, different committees that we've gotten help from along the way. We were thinking to sort of share with you some of the lumping and splitting issues that we come across. For example, when do you have, when you have a retina-only phenotype, and the same gene can also be responsible for a broader syndrome that has retinal issues as part of it. Like, is that combinable? Can we lump those together, or would they be better split into two curations? We're also going to talk about some disease naming issues that we come across all the time. This group has a particular preference a lot of times for dyadic naming, where you add the name of the gene right into your disease name, right? So that's just tends to be the norm in this GCEP. It would be interesting to hear about, GCEPs that you work for, and do they do things similarly or differently? And you can imagine when you're playing around with the disease terms that we have a lot of negotiating that we do and a lot of help that we get from the folks at Mondo. So we do a lot of requesting of parent terms and child terms and things like that. So I'll share a little bit of it with you as we go. I just wanted to say, as like a starter, that one of the strengths of this GCEP is that we have a lot of different experts, and they really there's a lot of geographic, I guess, diversity in the areas of the world that they work in, which is kind of a benefit when you're looking at a huge list of genes. So we have about 250 genes that are sort of the scope of what the group is trying to accomplish. And where these come from is the Genomics England panel app. I guess that consortium or whatever has like a list of what they call green genes. And so I guess in that kind of refers to genes whose relationship to the disease has been kind of rated as diagnostic grade. So that's sort of where we got our gene list from. Where we fit into sort of like a larger working group, this GCEP is part of the ocular domain. And so you can see here that there's a bunch of, I think there's 7 different ocular groups. And one of, I guess, the accomplishments that we could sort of brag about as a GCEP is that some of the genes that this group has curated have gone on to sort of be the subject of like their own VCEP. And so there are 3 different variant curation panels that are kind of sort of benefiting from the work of the Retina GCEP by taking it a step further and doing variant curations on those genes. Um, just to give you like a quick background, if you're like me and you don't have like research experience in sort of the retinal field, I'll just give you like a quick 2 minutes. Um, you could probably imagine that we're focused on inherited retinal disease. By the retina, they're kind of referring to like the innermost part of the eye, or they call it sometimes the posterior part of the eye. So the retina is like this pretty thin membrane, right, at the back of the eye. And some of the phenotypes that we see, they'll involve like different specific areas of the retina, like the central part. Sometimes people call it the macula, or the peripheral parts of the retina. And they can also involve some kind of like, we'll see phenotypes involving some of the adjacent tissues, like it's right next to the head of the optic nerve, or sometimes you have these blood vessels that like kind of come out of the retinal area. And so we see the phenotypes that we see can kind of involve all those different areas at the back of the eye. And although the retina itself is like this really thin layer at the back, it turns out that even within that thin layer, there's like multiple interesting sublayers, and all of those have to be kind of functioning properly. And so it turns out that dysfunction in any of these different layers can kind of result in like an inherited retinopathy. And you can see here, some of these cell types are going to be very familiar to you. Like in blue, these sort of like long blue cells are indicating rods, and the longer pink cells are representing cones. So you can see that the photoreceptor cells, rods and cones, are kind of organized into their own layer. And there's also this interesting sublayer that a lot of times is problematic in our patients that we're looking at, called the retinal pigment epithelium. So it turns out at the very, very back of the eye is this sort of like pigmented layer. And part of its function is to kind of absorb light that otherwise would be kind of bouncing around. And it also serves as kind of like a feeder layer or a support layer that kind of like is adjacent to the rods and cones and kind of like maintains them over time and secretes factors, that they need and stuff like that. So one thing that's interesting, that kind of like a trend that like has come up through our work, is that sometimes the genes that we're interested in actually like their products like localized to like really specific and interesting parts of the retina. So like and they kind of belong to like they group themselves into these molecular pathways that are kind of part of this light perception process, and also how how the eye recovers after perceiving light. So I guess you could say different subcellular localizations within these cells are kind of associated with different genes or different pathways. Um, Now, interestingly, like if you kind of look at the Mondo, like kind of family tree of retinal disease, it sorts itself into like 6 or maybe even a few more different categories. And you would think that these would be kind of like molecular categories, like all the genes that affect this cell type would be like in their own subcategory. But actually, they're kind of organized in terms of severity. Like this entity on the left side is really early onset, and then there's kind of a milder form of it that has its own branch in Mondo. And so they're kind of organized in terms of like phenotype and like what the presenting symptom is and stuff like that. And so you can imagine that our group has some interest in kind of reorganizing things to be a little bit more like gene dependent. or to have like everything belonging to one gene sort of organized in its own little branch. Um, so despite the high number of different, I guess you could call these like 6 different diagnoses that someone could get if they have retinal disease, some of the eye structures and some of the issues that are underlying them can kind of be in common. So like if you think about like early challenges that groups like ours encounter, um, one of them is kind of overlap with other, or I guess I could say one of the issues that we have is lumping and splitting, right? Like sometimes the same gene could be responsible for 2 different diagnoses. And we have to kind of undergo that dilemma about, do we combine them, or are they two clinically distinct? Um, that sort of triggers a lot of times if we lump together some kind of gene naming conversations, like I said, negotiations with Mondo and things like that. And then the third category here is like overlapping genes with other GCEPs, like when we can come up with like a syndromic case where there's retinal issues, but there's also like hearing loss issues, for example. Our group has kind of developed some procedures around, getting in touch with that other GCEP and sending them our kind of proposed name and seeing if it aligns with what they're looking for, or what they see from their perspective in hearing loss, for example. I did want to point out really fast that the group kind of, when it comes to lumping and splitting, you can see here that we discussed our strategies and got some pre-approval from the ClinGen lumping and splitting working group. So this is just like a quick ad. If you have any lumping and splitting issues, that group is tremendous and could be like really interesting to reach out to if you'd like to do that. One of the reasons that the work is kind of really important is that there's increasingly like therapeutic implications for certain diseases or certain, I guess, genes that are correctable by gene therapy. And so you can see here in this sort of like circular diagram at the top right, that there are increasingly available different sort of injectable gene therapies that can sort of get right to that back layer of the eye and sort of provide a gene that's missing and for like a loss of function condition. And so it's more and more important to have the gene associated with the phenotype, right? And so one example that we wanted to show you really fast is this gene called RPE65. It's recently gotten its own gene therapy. And when you look at its OMIM page, there's a few different diagnoses that can be associated with that gene. And it turns out some of them are amenable to being treated by gene therapy, and some of them are not. So that's sort of what kind of guided our lumping and splitting decisions a little bit. So here's like what the OMIM page here's what the Mondo kind of tree originally looked like for RPE65. It had a couple different diagnoses that patients can receive depending on case severity. If they're really severe, they would go down this left path. And if the cases are a little bit less severe, they would go down the right side. And also there's some differences you can see in inheritance pattern. So a couple of these diagnoses are recessive and one is dominant. Interestingly, we ended up lumping together the 2 recessive ones under this kind of umbrella category called RPE65 related recessive retinopathy. And the dominant one, we kind of split apart or kept it on its own for like a separate gene curation, right? And we also called those cases retinopathy, but we distinguish that they're dominant. One thing that this group generally does is kind of like preserve like those child terms, like to keep the same diagnoses underneath that kind of umbrella category. And we can talk a little bit more about that in a minute. Um, right. Um, so what are the benefits sometimes of lumping and splitting? One thing I wanted to point out, and I've kind of highlighted it here in yellow, is that sometimes you want to lump together the 2 entities that are treated the same way, right? So these 2 are going to benefit from that injectable gene therapy. And the third one that isn't highlighted on the right side is not going to benefit. It's sort of like a dominant negative kind of a mechanism kind of thing. And so, yeah, so one of the benefits, I guess, to considering lumping and splitting is that you can maybe merge a couple of terms that aren't so different from one another. Maybe the underlying variants are the same, or maybe their phenotypes are really heavily overlapping. Oh, thanks. Interesting question from Kristen. Did you split dominant and negative because the mechanism of disease is different? Great question. That's exactly right, essentially. Yeah, this group does a lot of splitting if you find that the mechanism of disease is distinct. Um, I don't know if you want to unmute Kristen, but feel free to if you want to. Right. So essentially, the mechanism of disease in this case kind of dictated that, yeah, some of these were going to be treatable and some of them weren't. Yeah. Another thing we wanted to point out really fast is that the field of kind of inherited retinal disease is kind of moving towards genetic diagnosis as as the model. And so they were saying the clinicians were telling us that the patients increasingly know their gene that's responsible. So they kind of are starting to identify, oh yeah, I'm an RPE65 patient. And so that was part of the rationale, I think, for including the gene name right in the disease, this sort of renamed disease. Um, right. Um, right. Oh, so I wanted to just address really quick why this GCEP, when we talked to Mondo about creating this term called RPE65 related recessive retinopathy, we didn't necessarily ask them to like obsolete the old terms. You didn't get rid of these diagnoses that, you know, clinicians still use and some affected families might still identify with these old terms. We didn't want to get rid of them. We just wanted to kind of make it clear within OMIM that those are sort of child terms underneath this sort of parent category. So that's one consideration that the group thought about. Um, Another thing I just wanted to like draw your attention to really quick or be transparent about is that this group we often have like lumping, right? And so we have to try to take two diagnoses that are a little bit different and think of like an umbrella term that would be inclusive of both of those. And so the umbrella term for this group a lot of times is retinopathy. It's like a very general way of kind of saying um, something's wrong with something's wrong with the retina. So it kind of unifies these two terms that share retinal features. Um, so yeah, I just wanted to kind of admit that in our first 89 gene curations, we ended up using the term retinopathy like 55 times. So it's always, for us, it's often gene specific or, name of the gene, then related retinopathy. And so just to kind of give you a sense of like what these two diagnoses are like that the group decided to lump together, they actually have a lot of really similar features. So the more severe form is in the middle of the table, the less severe form is on the right. You can see the biggest differences between them are kind of highlighted in yellow. Like the more severe form has earlier onset between age 0 and 5, and more severe vision loss earlier on. The less severe form can have, really severe vision loss can be anywhere up to age 40, and it has onset sort of later in childhood. But you can see why, even at the phenotype level, um, we thought that these looked pretty lumpable together. There's quite a lot of similarities. They both have like kind of rod and cone dysfunction. You try to get responses from the rods and cones, and they tend to be non-detectable or not much going on there. And they both have severe vision loss, like pretty early in life. Um, right. So I think I kind of already kind of covered this slide, so I'll keep it moving. I did want to ask this question, and I was curious if folks in the audience who work for a GCEP, would you mind putting your answer in the chat? Um, no pressure, but I just wanted to ask if you work with a group that, regularly faces lumping and splitting issues. I've kind of realized that some groups tend more towards lumping and some groups tend to default more towards splitting. I was just curious, would you mind to kind of like put in the chat, do the group that you work for, are you more of lumpers? Or are they more splitters? Um, Thanks for responding in the chat if you have a chance, just saying lump or split. Yeah, right after this, I'll kind of give you the answer for this particular GCEP. They sort of have their bias, and I'll kind of give you the rationale. Interesting. A couple people are saying 50-50. A couple of groups that tend to lump. Oh, that's really interesting. Yeah, okay, cool. Yeah, thanks for continuing to put things in the chat if you can. I feel the same way about this particular group, that they sort of tend towards lumping. There's like a little bit more lumping. And I'll kind of give you some background on how they reach that conclusion. The list of retina genes is actually quite huge, and it's like over 200, like I was saying earlier. And so far, we've done pre-curations for maybe 40% of the list, or maybe a third of the list. And about over 20 genes so far have assertions in the literature for relationships to multiple disease entities. So you can see within this pie chart, maybe 75% of our list is just associated with the one, and then about 25% of the time we have to think about lumping and splitting. And the disease entities can get pretty complex. They can go up to like, 6 or 9 different things you have to consider whether to lump them together or split them apart. When it comes to, there's definitely some interesting trends that come out when you look at our inheritance patterns. A lot of our genes are they're either recessive in blue or they're dominant in red. But you can see there's this weird purple zone where some genes are associated with both recessive and dominant diseases, and that gets really weird, that lumping and splitting. So one thing I should tell you off the bat is that a lot of the entities that we look at have the same mode of inheritance. And we've noticed that those in particular tend to get lumped. So I'm curious if that's a trend for you all too. Like those are people that are from lumping groups. Maybe you're lumping across the same mode of inheritance. I don't know. This is just kind of a summary of what we usually find. So each gene that we've looked at is kind of a different row in this table. And you can see that there's a whole column that highlights the modes of inheritance. And a lot of them that are kind of have multiple recessive forms, for example, we add them up into one, I guess, gene-specific retinopathy. So if you look at the top of the table, we've lumped all the recessives into ABCA4 related retinopathy. And so we came up with like 15 different examples of multiple recessive forms of the same gene, and we lumped in all 15 cases. We didn't find it was that consistent with the dominant. It looks like at the bottom of the table, you can see like 3 out of 4 of the dominant ones all got lumped together into one dominant disease. And we generally called that, for example, GUCY2D related dominant retinopathy. But there was one exception where we found 2 different, like clinically distinct dominant disease entities. And similar to the point that somebody brought up earlier, I think it was mechanism in that case that was a little bit different between one dominant that was kind of haploinsufficiency and the other dominant, which was more of a kind of like a dominant negative or something like that, or a gain of function. Now it got, like I said, it got really weird when you have multiple modes of inheritance. You have like dominant and recessive. Do we lump them together and make it semi-dominant? That was always a bit confusing for us, and those have been really long conversations sometimes that this GCEP has had. We'll just sum up in this table. It's kind of summing up some examples of like AR disease entities that are shown in the first column, dominant disease entities that are shown in the middle column, and then you can see in this column, you can see the decision. And it looks like we split almost all of them. In most of the cases, we found that the recessive and dominant curations had different mechanisms, right? They're caused in by different variants, and they show up in different families. There's an interesting question about triallelic or trigenetic inheritance. That's an interesting question. I think we've talked about it a couple times, but I don't know if we've ever curated anything for that. Could I ask a quick follow-up question to that, Ramakrishnan? Could I ask you, does that refer to multiple different genes involved, or does it refer to like 3 copies of the same gene?
Years ago, Harvard Dryga and others has a series of papers published, and in fact, RP, there are 3 or 4 diseases, 3 genes are needed. It is now in a standard textbook is also it is accepted. In fact, 3 genes are needed, not 1 or 2, 3 genes to show the disease phenotype. And it can be on cord or cord, etc. But I forgot the things. But I believe that it is a standard accepted in genetics now.
I would say we've been increasingly thinking about it. Like we've increasingly, been looking at one gene and realized that some of the cases are at least digenic. I think your example is really cool, and I need to research a little bit more.
It usually form a complex because in the complex formation, 2 or 3 proteins has to assemble it together in order to get to the proper structure. Any one of them can disrupt, the mutation can disrupt the functioning of the gene. And it is well defined, well known about, I think 15 years ago, first paper is published by Dryga and colleagues, DRYKA.
I think that makes a lot of sense. It's completely believable, right? That they're forming a complex together and variants in multiple genes can kind of interact to give you the phenotype. Could I ask you, have you curated any of those genes in
No. But that is from my remembrance, because even I teach that trigenic inheritance to undergraduates and medical students.
Yeah. I think that's super interesting and a little bit cutting edge for us, you know. Thank you, Christy, for confirming in the chat. Yeah, I don't think that our group has curated any of those yet. You know, I maybe I'm trying to remember, but I think one of the kind of maybe weaknesses, I guess, of the ClinGen gene curation protocol is it is not very well tailored for like multigenic things. You know what I mean? So we would kind of be evaluating one of those three genes that you're talking about, but with it keeping in mind that the other two genes might be part of the real picture, right? Yeah. Yeah, that's a great question. Thank you, Christy, for putting interesting stuff in the chat.
I was gonna say, I'm not really the right person to comment on that, but the gene curation framework is designed for monogenic inheritance, which, like as you said, it's a weakness of it that it can't do things that aren't strictly monogenic, but that is what it's designed to do. But I do know that there is I think a complex inheritance task force that's been started within ClinGen that is going to be coming up with some ways that we can be assessing these digenic, trigenic, or complex mechanisms. So that's space to watch in future.
You are talking about retinopathy, but the phenotype will be the same. And perhaps curation has to consider that, you know, even though it is monogenic allele, then what about autosomal recessive, compound heterozygote? So we can go on taking a lot of them. We are not strictly doing monogenic. In fact, each one is a separate gene to some extent. So, but assembling or something, it is producing the disease phenotype. So that is more important, I think. We cannot completely say that everything is single gene. Usually, retina, a single gene is a complex, extremely specialized organ. It requires multiple gene anyway for formation of any of the things we are talking about. That I can understand the complexity. I don't have any answer. I don't have any solution. So I am not in the field either. So only old remembrance of curiosity's take only I ask.
Yeah.
Jump in for a second. Can you hear me, Bill?
Yep.
Oh, yeah. So yeah, I think this is true of a lot of fields where there's a high locus heterogeneity, like hearing loss or seizures, ciliopathies in general. And you're back of the envelope, if we do exome sequencing, we're going to find 100,000 changes, which works out to be about 5 changes per gene. So benign and pathogenic together. So when we're sequencing these sort of 500 gene panels for patients with retinopathy, we're always finding, besides what we believe is a primary or core gene, which could certainly have modifying effects from other genes, which you could argue that's oligogenic. But besides the core gene, we often find that the person is a carrier for one or more other autosomal recessive disorders that do relate to the retina. So, I think we either need really, really enormous populations, or we need really, really well curated families with broad sequencing of all affected individuals. And I think those are still, in the works. There's certainly some case examples, but the replication and validation of like a triad of or even in some cases a dyad besides the PRPH2 ROM1 are not often replicated. But I think these are great discussion points.
I appreciate what you are talking. It is not a modifier. It is completely a gene is involved completely. And a lot of literature is I am not in the field. It is not a speculation. It is shown sequencing by many groups. Literature is about trigenic inheritance. So naturally, it's a well-known fact. It is the gene is not a modifier or susceptibility or something. That is, I have to tell that it's not a modifier. It is a functional. 3 genes are needed. 2 genes, if it's there, it won't have any problem. So another 3rd gene is needed for that. It can behave almost like autosomal recessive type where 2 alleles have to go something like that. So the mutation has to be there for 3 genes. So
It's triallelic.
I have to make sure that you presume that it is a modifier. It is not modifier attenuating gene expression or something. It is very different.
Sure. I guess at least in the eye field, there's triallelic digenic. So we've seen that with two of the achromatopsia genes, CNGA3 and CNGB3. And I think Nikos Katsanis tries to make the case about Bardet beetle having potentially multiple genes involved. But again, I think if we're thinking about things that can be replicated in more than one individual or a small family, we haven't quite seen that level of evidence that we could get trigenic up to definitive in the ClinGen framework. Again, because of the limitations, the way we think about core gene versus the core genetic causes and things that we can really distinguish from being a modifier effect.
You can search in PubMed Trigand for this. They have a lot of papers well characterized by sequencing, and it is well established. So it is not. If anybody do a PubMed search, you will find out enough papers well established. So it is not a speculation as presumed to be. That's why I'm going to say. But again, I am not in the field for 5 years. I am not following this. So naturally, I am completely a lab right now. So doing diagnosis only. So I am not going out anything further than that. But I know that it is there.
Yeah, I think it'll be really interesting to see, maybe what the outcomes are from this sort of task force that's going to tackle this complex inheritance question, and maybe to find out what kind of modifications to our framework needs to happen in order to, kind of document, like you said, this kind of emerging trend. That's really cool. Nice.
Thank you.
Yeah, thank you for asking a great question. I had another question for the audience, if you all don't mind. Since I was kind of showing on this slide that we tend to split, at least the group that we work for tends to split a lot of times recessive forms from dominant forms of the same, when the same gene is responsible, with a couple different exceptions. So a couple times we've had a semi-dominant mode of inheritance, but we tend to split under this condition. I was curious if this is something that you've ever come across in your group. Could you put a quick note in the chat about whether you've lumped under those circumstances, or have you split under those circumstances? In other words, do you ever curate anything as semi-dominant? Thanks for responding if you have a chance. Yeah, I'm not sure if this is one that people have come across a lot of times. Cool. It does look like there's some examples. Yeah, I agree, Jenny. It's sort of like yes, but rarely. I really like this point that Allison has brought up in the chat, that you said that you split in most cases, but you think semi-dominant in cases where they tend to share variants. I think that's a really interesting point. That maybe you're getting at like mechanism as well, Allison, right? Oh, interesting. Thanks, Christy, for mentioning this. So sometimes they lump and do a lot of semi-dominant things in the hemostasis thrombosis field. That's cool. So I was going to walk you through, if you don't mind, I'll kind of walk you through like an interesting example of a super complicated lumping and splitting gene that we got for the retina group. And if you don't mind, like, please unmute at any time or put interesting things in the chat at any time. It's not so critical to get through all the slides, but just to have a conversation. So we visited the OMIM page for this gene, GUCY2D. We found that there was a kind of a combination of dominant diagnoses that these patients can receive and recessive diagnoses. So there's a lot going on there. We first I'll give you like a little background. This is a gene that kind of dimerizes. It's kind of like a membrane protein that functions in the phototransduction cascade. The dimerization. And start out by considering like all just the recessive entities, right? There are 3 different recessive diagnoses. They're kind of asserted to be different diseases in the literature. So that's sort of, you're probably familiar with this sort of diagram that ClinGen folks use, right? With all the factors on the left leaning towards splitting and any evidence on the right side of the screen pushing you to maybe lump them together. So we found that because they were all recessive, that was sort of like one kind of underlying reason maybe to lump. But we also wanted to look at molecular mechanism. In other words, do they share any of the same variants? Do they run in the same family, these diagnoses? And we actually found if you do like a Venn diagram, and on the left side of the screen are all the biallelic cases, there's really no overlap in these Venn diagrams. And if you sort of like look, if you kind of lay all the variants along the top and the bottom of the gene, basically the ones in blue receive a diagnosis of Leber congenital amaurosis. The ones in purple, they're associated with a diagnosis of congenital stationary night blindness. And the ones in red are associated with cone rod dystrophy. There aren't any like strong patterns or like concentration of any of these variants in a specific area. And if you look at, a lot of them have an X at the end, indicating that there's a stop codon in there somewhere. And so it kind of makes you think these are all loss of function variants, and they're all showing up in a biallelic state. So really, what we thought we were looking at was basically 3 different diagnoses for patients who have 2 loss of function variants in GUCY2D. So we felt that there wasn't like strong evidence for a mechanism difference. We did want to look at the phenotypic variability between them first to see like, do those 3 disease entities, are they distinguishable in the clinic? They certainly overlap quite a bit in their phenotypes. On the left side of the table, you can see that they're all kind of limited to the eye, and a lot of them kind of, they exhibit some kind of issues with basically reduced visual acuity. You have vision loss in all these states. The rods and cones have abnormal responses. They kind of tend to be on the reduced side. You can imagine, like, since rods are kind of keeping your night vision good, patients tend to have night blindness in a lot of these conditions. Since the cones are responsible for the color vision, that's abnormal or frequently problematic in all these conditions. So we felt that there was quite a bit of phenotype overlap. And so we ended up thinking that the evidence was accumulating on the right side of the screen to kind of lump together all these recessive conditions, but they're loss of function, and they tend to be like have similar retinal problems, I guess. On the other hand, like looking at the recessive versus dominant disease, that's where it got really complicated for this particular gene. I guess the recessive and dominant diseases sometimes are diagnosed with the same, either clinically can receive the same diagnosis. But on the other hand, their inheritance patterns are different, so that's kind of a motivation to maybe split them apart. We did want to look at the variants. Like, in other words, is it possible for like a family with recessive disease to have individuals with just one variant that have dominant disease? Do they run in the same families, these recessive and dominant cases? Or are they like totally different variants? So we arranged the variants again, like along the length of the GUCY2D gene. Along the bottom here are shown the autosomal recessive variants. You can see they show up lots of different places along the length of the gene. Interestingly, all the dominant variants that we could find were localized to this DD domain. That's kind of the part of the protein that mediates dimerization. And so it sounded like maybe these are very specific variants are dominant, and they're probably dominant negative, right? They're disrupting the ability of the protein to form dimers. And so it kind of looked, to our GCEP, it looked like these are 2 different mechanisms of disease. One of them dominant negative, or kind of disrupting dimerization, and the other one recessive, kind of loss of function. So that was kind of another strike, I guess, on the side of maybe splitting them apart. And where it got really weird is taking a look at the phenotypes. They weren't that different. We wanted to know, could you distinguish them in the clinic? And so I did ask the experts. They said that you can distinguish the dominant forms from the recessive forms in the clinic. You can see that there's some rows for phenotypes that show up in one but not the other. Some of the more severe phenotypes show up only in the recessive form, which is a bit of a more severe disease. And the dominant form tends to be cones more than rods. So you could imagine that color vision and parts of the retina that are cone associated have a little bit more detail there that are kind of unique to that disease. But overall, you couldn't say that they were completely clinically separatable. So we ended up saying that the phenotypic variability was kind of a reason to lump, maybe. These were kind of clinically not that different. While a molecular mechanism was kind of pushing you to split because they were running in different families and caused by different variants. And so, yeah, the evidence was really was really even on both sides of things, so lumping and splitting. So I was wondering to ask you, this is sort of like a poll question again for the audience. This question at the bottom, when you find that there's a combination of kind of things pushing you to lump together and things pushing you to split apart, which piece of evidence should be like the deciding factor, And I think the answer to this is different for different GCEPs. But I just wanted to ask for the groups that you work for, or maybe for your personal opinion, do you kind of vote for A, B, C, or D? Like, what should be the tie-breaking piece of evidence that has a little bit more weight than other things? Thanks for responding. If you have a chance to just put like A, B, C, or D in the chat. No pressure, but thanks if you have a chance to think about this. Interesting. A lot of people voting B, a lot of people saying molecular mechanism. Some folks are kind of saying inheritance pattern can be a really good clue as well. You're definitely thinking along the same lines that our GCEP tends to think along, I would say. I do think some other folks kind of go with phenotype. So I haven't seen anyone vote C yet, but that's interesting. There's no wrong answer as far as I know. It tends to depend group to group, right? But I'll kind of give you some of the reasons why. You can see at the bottom of this screen, our GCEP ended up recommended splitting in this case. And we did that from a combination of looking at the inheritance pattern, but also, like, I think more than anything, looking at the mechanism. The patients with the loss of function variants were all over the gene, and whereas the dominant variants were all kind of restricted to this one domain, it just looked mechanistically distinct. Cool. Did anybody want to weigh in here or talk about, your answer and how did you feel different at all? Or please feel free to unmute or put more detail in the chat if you'd like. Yeah, no pressure, but just interrupt me at any time. It should be totally fine. Right, so as I mentioned, our group ended up recommended splitting into 2 separate curations. So we ended up curating the dominant disease entities all got kind of pushed together into one category called GUCY2D related dominant retinopathy, whereas the recessive cases, we pushed those together into a parent term, or sorry, underneath a kind of an umbrella term called GUCY2D related recessive retinopathy. So in both cases, we wanted to have this general term for retinal disease, but we distinguished them based on the inheritance pattern. And right. I was going to get into like another topic a little bit, if that's okay. We were going to talk a little bit about
this is Mandy. Can I ask a question related to this? It's come up as a clinical query. When you have different types of variants that are causing different diseases, and you've now decided they're different entities, so got a different gene related disease, can you, firstly, what happens when you don't know the mechanism? So you just got the variant and you haven't proven it yet, particularly if it's not all in one domain. I'm assuming you have to curate against each disease to decide which one it's going to fit in. And if you don't know, then maybe it's VUS for both. But what happens once you have proven, I suppose, that it fits, it's pathogenic for one? Does that mean, and this has really come up as a question, that you can use, say the phenotype is distinct enough that you can say, oh, it's fitting the dominant negative one, it's now pathogenic for that. Can you use the fact that it occurs in people without that phenotype as evidence against pathogenicity for the other gene-related disease? I'm not phrasing it very well, but it's almost like you've seen it with the one phenotype, and maybe it's even not enough to become pathogenic for that other phenotype. But can you know how do you treat it? Do you just ignore the second disease entity and don't curate for that? Because that gets very confusing, because you've got the same gene, same variant, and then it would almost have to be curated. This is for the variant curation, be curated twice and entered twice. For one, it might be pathogenic. For the other, it's definitely not pathogenic because it's causing the other disease. How is ClinGen and ClinVar going to handle that? Because I think it's really very confusing for the clinicians. I actually have to find the query that I've got, because I've been ignoring it for 6 months because it was too hard for me and thinking. But it has come up before, particularly, I think before, this would be prior to any gene curations being done. People are dealing with these right now.
Yeah. I think that's an awesome, Mandy. I'll just give you my quick.
It's RET. RET is the example that came up.
And so was there a variant that was seen like in the...
And the other ones are that came up were POL-E, which is definite one, where it's recessive for, say, truncating variants in a nasty disease, but a very few dominant negative variants exist to cause dominant cancer. And SMARCA4 is the other example that were where you have distinct phenotypes for gain of function versus loss of function.
Okay. I mean, this is just, I'm a total non-expert in the variant curation side of things, Mandy, but I think this is an awesome question. So my kind of dumb answer of what I've seen in very few examples is that the groups tend to curate for whichever of the two assertions is more convincing. So if you had like a RET case or something, I think they'd probably, you know what I mean? They would guide it towards that disease mechanism. Do other folks feel, or what do other people do in variant?
I have a question. So when you are talking about autosomal dominant, it will come penetrance and skipping of generations. So one generation, it may not have any phenotype. Okay. So how can you lump and this procedure will be done if a family study or generation is not done, for study is not done? It is notorious. Most of the autosomal dominants have some kind of penetrance. In some cases extreme, it may skip the generation. One generation may not have the disease, but they have the mutation, but it expresses in the next generation. So it's a perpetual problem for genetics, but even after all these ClinVar and other things, or DNA sequencing and other things, these fundamental questions are not still answered.
Yeah, that's a good question. That's another good question. I think penetrance is definitely like a big problem too. Yeah. Thanks.
I am saying in extreme cases, penetrance in such a way that there won't be any phenotype. It just skip generation. Many cases.
So back to Mandy's question, there are definitely examples of that where, like, if it's a loss of function versus a gain of function mechanism for two different diseases, that sort of by virtue of what the mechanism is, if something's pathogenic for one disease, it's going to be benign for the other. We don't have too many examples of that within ClinGen at the moment where that's come up. RYR1 is definitely one of them. So with malignant hypothermia and the myopathy phenotype. But I don't know that disease area super well. I know there are definitely some variants where it is pathogenic for one and benign for the other, but that's not always true. It might be pathogenic for one and a VUS for the other. I think there's some uncertainty around the mechanism for one of them. Again, not entirely my area, but there are definitely ones pathogenic on one, benign on the other. And at the moment, that isn't well handled by ClinVar. So both VCEPs within ClinGen have submitted those assertions to ClinVar. And at the moment, it flags as a conflict because they're different. So ClinVar at the moment doesn't separate out based on what the assertion is for the disease entity. It just looks at it as a whole. That is an issue that the ClinVar team is aware of and are working to try to come up with a way that can be better teased out. So it doesn't
it's
properly answer your question, but sort of gets at
it. It's I guess a process. Because at SMARC-A4 that was asked of me is whether if you can prove that it's not the one disease mechanism and you've got the patient with another phenotype, which is why they were tested for the gene, of course, but you can see that it's not loss of function, you should be able to say it's benign for that presenting patient. And I know this gets very tricky because people still often curate using the patient's phenotype as what they're going to choose as a gene disease relationship they're curating for, even though in my mind, you should be curating for all possible gene disease relationships for a given gene if they exist, and then deciding which ones you would put in. But Pol E, certainly it's come up to me where something was flagged as pathogenic in a colorectal cancer patient, and it was the truncating variant. It was for the recessive disease. And I had to chase it up with the company too. Because I thought, what? Have they changed mechanisms? So yeah, I guess it's, but maybe flag this as something to discuss because it's certainly come up. I've just been ignoring it because too many other things to do.
That idea of curating a variant for all possible associated disease entities is also an idea that will be touched on more formally in the next version of the SVC standards as well. But just in the interest of time, we won't go into that any further. We have 5 minutes left, so I would hand back over to Bill and see where we get to.
Mandy, I was just going to direct your attention really quick to the chat. Christy had a cool reply to you that makes a lot of sense also. Not to steal her thunder, but just to say, or I don't know if you want to unmute Christy, but you had an interesting answer to say if you had a benign conclusion and a pathogenic conclusion for the same variant, but for different disease entities, maybe you would only submit to ClinVar the pathogenic one. That seemed interesting.
Yeah, we are a group where there's a gain of function phenotype and a loss of function phenotype, and it's always going to be if it's pathogenic for the dominant form, it's usually VUS for the loss of function recessive form. So it just confuses people when it's put in twice and it's put in under different classifications in different diseases. So we've decided to, right now, we don't even have rules for the dominant one. So that's the other issue. You might not have rules to cover all of the phenotypes. You might just have rules to cover one specific phenotype, and therefore you need to stay in your lane and just curate for what your rules were set to curate for. So our rules were set to curate for the recessive loss of function phenotype. So we avoid variants that are gain of function at this point until we have time to write the rules for those. But having multiple assertions in under your expert panel tag, I think it's just confusing. So maybe trying to stick to whatever you think is that variant's pathogenic for is probably the most helpful. You can always write something in your variant summary that gives, some more explanation to say this would be VUS for the other phenotype or whatever, and that way you've covered your bases.
Alicia has made a very good suggestion on one disease called malignant hyperthermia. It can just behave differently. Is again, finally, everything depends upon the amount of the protein or enzyme is needed. In fact, malignant hyperthermia permission is almost behave, even though sometimes it is considered to be autosomal recessive. If 50% of the protein is needed for that, then it is okay. But if it is less than, that will not. Even autosomal recessive disease, except metabolic disease, truly there is no autosomal recessive disease. There is phenotype is amount of the protein which is enzyme it is produced basically. So there are gradation of phenotype will be there. I perhaps going too much into this, but still we have to understand that finally molecular mechanism is the amount of final protein is there. If more proteins are produced, a mutation in which if you get more protein or enzyme, perhaps it will be alleviated even in the autosomal recessive null alleles are there. In the case of autosomal dominant recessive, 50% may not be needed. So all these things is become very complex in genetics. So here we are just putting into a separate compartment, lumping and this one. Perhaps there are some difficulties you may find out, and then bringing that to disease phenotype. I don't have a solution, but it is we are making a watertight compartment, putting one box, two box, three box, and mark them. That I don't know. It is genetics are not intended to that. That's my understanding.
Okay, great. This has been a really interesting conversation. I hope you don't mind. I'll just do a super fast wrap up. Right. I think we've kind of gotten through like pretty much all the slides I wanted to show you all. I just wanted to like stop at the end really fast and say thank you so much to all the people whose work went into the presentation, and thank you all for the great participation. If you have other questions or you have other interesting points you want to talk about, just like get a second opinion, please feel free to email me, and thanks so much.
And thank you so much, Bill. I always personally really enjoy whenever we have you on to present, and I learn so much and love how you manage to inspire such discussion amongst all the people on the call, which is really what this forum should be about. So thank you for, again, a wonderful presentation. As I said at the start, we won't be meeting again at the end of this month, but we will be coming back in February on the 14th, I think it is, for the first one. And we'll be having sort of a little mini series of sorts on Nomad V4. So should be excellent learning opportunity and excellent discussion opportunities as well. So keep an eye out for some more details for what that's going to look like.
Wonderful presentation, William. I enjoyed very much. Please keep it up so that we will learn more.
Thank you so much. Nice
presentation. I appreciate that. You've done a very good work.
Thank you all.